Hello, everybody. And yeah, thanks for attending this talk. I'm sorry I can't make it in person today, but I hope you enjoy my talk anyway. So today, I want to outline my thoughts about a topic that is a little bit away from the normal research questions we have with biomimetic robots. But it's very important because when you work with biomimetic robots, you always have to work also with live animals. And involving live animals in experiments requires a very careful planning, and you have to take into account that the animals don't suffer, that you don't use too many animals, and that you try to avoid using animals where it is possible. And that is all subsumed under the 3R's principle, which means that we have to reduce the number of used animals to an absolute minimum, that we have to refine our methods so the animals don't suffer during the experiments, and where possible, also exchange animal experiments with experiments that don't involve animals. And I want to outline how biomimetic robots will help to promote these principles. But at first, I will show a little bit what kind of biomimetic robots I will talk about. And I will talk about fish-like biomimetic robots. Here you see me presenting our Berlin Robofish, which is a guppy-like robot. It's a dummy fish that we print with a 3D printer, and then we color it, and we put a magnetic base on that fish so we can couple it with a robot unit that is driving below the tank and steers so the dummy fish. Here you see it with a couple of guppies interacting, swimming in a tank. But there are also other forms of fish-like biomimetic robots. We have robots that are attached to the robot base or the robot unit at the top. And this gives you much more freedom to move the dummy fish in 3 dimensions, for example, which is not possible with a robot where the robot unit is below the tank and where dummy fish is attached with magnetic bases. But there's also the possibility to put all the robot unit parts inside the chassis of the dummy fish. And then you have bigger robots, of course, but you have much more degrees of freedom to move that robot. And some examples are, for example, the zebrafish-like robot that Maurizio Porfiri is using at New York University. Then we have the fighting fish-like robots that Donato is using. Then we have our guppy-like robot. And we now have at University of Constance a koi carp-like robot. All these animal-robot interactions are very nicely reviewed by Donato and Cesare in their 2019 or 17 review, and I really recommend reading this review to get into the topic of animal-robot interactions. Okay, so a first requirement for biomimetic robots to be able to promote the 3R principle is that these robots are accepted as conspecifics by live fish. And we found out that attaching little glass eyes to the dummy fish is really making the difference. And with these glass eyes, our live guppies followed the replicas, the dummy fish, comparable to what they do with a live conspecific. So in which modes can we steer robots? And of course, not all robots could be steered in the same way, but there's some general patterns. For example, we have the open loop control mode, which means that the robot is steered on a fixed trajectory that we pre-program into the software, and then the robot is executing this specific path. It looks like this example where the live fish leaves the refuge and the robot is then programmed to just swim on a zigzag trajectory through the tank. We use this experimental design a lot for our stuff because it is really yeah, it helps us to investigate the individual differences in the following behavior of the guppies. In this case, of course, yeah, you see a very social guppy that follows very closely, even when it's yeah, almost hit by the robot. But there are also examples where the robot is not followed by the live fish. Okay, and the second important steering mode is the closed loop control mode, which means that the biomimetic robot is interactive. So it reacts more or less in real time to the actions of the live animals. And that, for example, can be done by incorporating the cousin model, a zone model into the robot software, where the robot changes its behavior based on the distance to the live fish. And we have an example here. Here our robot was programmed to lead the live fish, but only when the live fish stays close to it. When it's not following the robot was programmed to come back. So as you see here, the live fish is too far away, the robot comes back. But now the live fish is more or less convinced to follow the robot, and so the robot is steering through the tank and approach some random goal points in that tank. And we increase the speed because of the video, because this following behavior now lasts for more than 10 minutes. Okay, so now we know what kind of biomimetic robots for fish experiments are out there, and now we'll show how they can really help us, for example, to reduce the number of live animals in testing. And my first example is a classic mate choice experiment. And here, dichotomous choice tests are used, which means that we have a focal individual, here, this individual, and we want to know the mating preferences of this individual. And we present two live stimulus animals at both sides of the tank, and we measure the time the focus individual spends close to these stimulus animals. And then, yeah, it has a preference for the one where it spends most of its time. In a very recent experiment, Donato and Cesare used a similar dichotomous choice test, but exchanged the stimulus animals with biomimetic robots. They asked whether guppy males have a preference for a pregnant or a receptive robot model. And they changed their replicas to be receptive or pregnant, but just adding a big belly. And a big belly means pregnant in these live-bearing fish. And yeah, of course, they only used one live animal instead of 3 live animals because the 2 stimulus animals are now artificial. And it works. So the prediction was that guppy males will spend more time next to the receptive model, and that is exactly what they found. Similar, we can we can look at shoaling experiments. It's the same dichotomous choice design, but not with stimulus fish at both sides, but at one side you present a shoal of live fish, and then you count the time the focus individual will spend close to that shoal and how much time it spends away from the shoal. That gives you a measure for how social or this animal is, or what kind of shoaling tendency it has. Using an open loop controlled robot can do the same. We can measure how much time the live fish spends close to the robot that is swimming inside a tank. And here we just have, again, our focal individual and no stimulus shoal. Whether it be a shoal of 3 individuals or 5 individuals, doesn't matter in that case, but we can really reduce the number of tested individuals. And as I showed you, the zigzag trajectories, they can also help us finding individual differences in the social responsiveness of live animals. And that's because we can present live animals with very standardized social cues. And this standardization that is very beneficial for us and for the animals. I will show you why. So presenting these standardized social cues also works, for example, when we want to test different phenotypic appearances of the dummy fish. We can all steer them with the very same behavior. So there's not a body size dependence or a color dependence of the behavior of the robot, which also standardizes the social cues presented. We can program the robot to just follow the live fish, also using the closed loop behavior to present live fish with very standardized social cues, which is not possible with live animals. We did it here in a recent paper showing that the speed of the live fish has a huge impact on the collective behavior. And we could show that, yeah, our robot can follow very closely the live fish movement. And this very standardized social cues, they reduce the signal to noise ratio in the data. And thus we can decrease the sample size to the very minimum of what is needed to find the given effects. And reducing the sample size obviously also reduces the number of used live animals. Okay, the next point is that biomimetic robots can reduce the handling stress for the fish. Here is an experiment, a very nice experiment by Christos, Iano, and Ian Cousin, where they trained some individuals to be the leaders, to always swim to the left or the right corner of a tank, and they tested these leaders within a group of otherwise naive individuals, and they asked whether the naives followed not in their test, depending on whether the leaders swam straight to the corner or whether the leaders were more meandering through the tank. And the point here is, besides it's a very nice experiment, a very nice data, very nice results, the leaders had to be trained a lot to really swim to the respective corners. So the leaders underwent extensive intensive training to swim to the desired location. And this training involves handling, and that handling can be stressful for the individuals that have to be trained. By using a biomimetic robot as a demonstrator, as a leader, we don't have to train any individual. We can have many goal areas where the demonstrators swim to without any training. So we just observe what the naive individuals do without having extensive training of the demonstrators. And we can do that with an open loop or a closed loop design, as we have here, where we programmed the robot to swim to a specific corner in the tank based on the Cousin model, and it works as well. And we find the same splitting of the following tendencies as Christos and Ian found. So here, when you look at the distance to the Robofish, in our case, it really depends on the direction of the motion. So how straight the robot approached the corner with a very straight movement, had a lot of fish don't follow, but at the same time, several fish also following. So the groups are splitting. And this is exactly what Ian Cousin and Christos Iano found in their paper with the trained demonstrators. Okay, and my last example how biomimetic robots reduce handling stress, it's a very obvious one. In that paper here by Rob Heathcote and Darren Croft, they looked how guppy predators approach their prey. And in prey-predator interactions, there's, obviously one that suffers most, that it's a prey item. But they exchange the guppy prey by a biomimetic robot. So they have a moving guppy model that is attacked by the predator. And this way, no guppy was hurt during this part of the experiment, and they could have the same quality of data, seeing where where the predator is attacking the guppy. They could color the guppy differentially. They could change the eye size of the guppy, and they could do a lot without hurting any guppy. So that is obviously reducing the suffering of a live animal. So yeah, let's come back to that sheet and look how the 3R principle can be promoted by the biomimetic robots. Can we reduce the number of tested animals? Yes, I showed how it works because we can exchange some of the stimulus in the woodworks by biomimetic robots. Can we keep the suffering low? Yes, we can, because we can reduce the handling stress, and in predator-prey interaction, we can exchange the prey with a robot or a dummy. So can we replace animals completely in our experiments? Well, for my research, that's not possible because I'm interested in the behavior of the focus in the woodworks, of the focus life in the woodworks. But for different questions, we can do so. Look at the koi carp-like submarine robotic fish at University of Constance. They exchanged all the live animals with their robots and looked how the swimming synchrony affects the energy demand during the swimming. And they didn't even use a single live animal in their tests. So even replacement is possible with biomimetic robots. And yeah, I want to close my presentation by emphasizing that this kind of research is only possible when you combine the knowledge of different scientific fields. And this is a picture of our group at a sulfidic spring in Mexico, where you see biologists and physicists working together on the same questions. And that is the same with the robot, with biomimetic robots. You have to combine the knowledge of biologists, physicists, engineers, and so on to really answer your questions. Okay, thank you very much for listening to my talk. Here is a list of some of my collaborators, and I'm especially thankful to the PhD students that are involved in the Robofish project. It's Juliane and Lea and Fritz. And yeah, thank you very much. If you have any questions, just write me an email and we can discuss this on that way. Thank you. Goodbye.